All right, so now we're actually going to start looking like we're learning about anatomy and physiology. We're not quite to the organ systems yet, but we are going to talk about tissues this week. So there are at least 50 trillion cells, and we said they can come in about 200 different types. But those cells really only form four categories of what we call tissues: epithelial tissue, connective tissue, nervous tissue, and muscular tissue. So above tissues, we have the organ, and the organ is a structure that has its own boundaries, but it's made of two or more tissue types. When you look at tissues under the microscope, we call that histology, and how they are arranged into organs. And we've already seen some tissues in some of the lab work that we've done. So a tissue is a group of similar cells and cell products. They work together to perform a specific role in an organ. Again, there are four primary tissues: epithelial, connective, nervous, and muscular. They differ from each other in their types and functions of their cells, the characteristics of the matrix, which is the extracellular material around the cells, and the relative amount of space occupied by cells and or matrix, kind of the ratio. That matrix, is the extracellular material, is composed of fibrous proteins, a clear gel called ground substance. It's also known as tissue fluid or extracellular fluid, or very often I use the term interstitial fluid. It has water, gases, minerals, nutrients, waste, and hormones. When you are looking at sections doing histology, when in the microscope, you have to be aware of how they were prepared and some of the things that can happen, what we would call artifacts. So histologists use sections, tissues sliced into thin sections of 1 or 2 cells thick. They have to be fixed. They have to be try to freeze them in the moment. That's generally done with something along the lines of formalin. They also have to be stained. Color doesn't naturally exist, so those different stains bind to different components in the cell. And both fixing and staining can introduce artifacts to the process, to the slide. So sectioning reduces the three-dimensional structure down to close to two-dimensional slices. Longitudinal section, cross sections, transverse sections, those are same way we used them in the beginning of the year. An oblique section also. And we've looked at these before. We looked at a whole organism. Last time we looked at planes and sections. Now we're looking at an individual organ. And that individual organ would be a very thin section that we could put under the microscope. Some tissues don't have to be sectioned. We can do a smear for things like blood cells, or a spread if you just lay it out on the slide, and you get a cobwebby tissue. Areolar tissue can be used that way. Epithelial tissue, the first type of tissue, are sheets of closely adhering cells, 1 or more cells thick, with the upper surface exposed to the environment or an internal space. These are linings and coverings. They constitute most of the glands. They are avascular. They don't have blood vessels, and they are generally nourished by the underlying connective tissue. The functions of these linings and coverings, epithelial tissue, is what you might expect for a lining and covering. Protection, protects deeper tissues from injury and infection. Secretion, some of them produce hormones, release mucus, sweat, enzymes, and other substances. Excretion, there are places we can get rid of waste from the tissues. On the other side of that is absorption, absorbing chemicals such as nutrients. We can use them for filtration, so they are selectively filtered by an epithelium. And we're also important locations for nerve endings, so they are involved in sensations. The general structure of epithelia is that cells are close together. There's very little extracellular material. Again, they are avascular. They have no blood vessels. They are nourished by the underlying connective tissue. They have a high rate of mitosis near the connective tissue layer that is supplying the nutrients. They rest on a basement membrane, which is a layer between the epithelium and the underlying connective tissue. That's made of things like collagen, glycoproteins, protein carbohydrate complexes, and it helps to anchor the epithelium to the connective tissue just below it. The surface of epithelial cells can be a basal surface, an apical surface, or a lateral surface. So if it's a basal surface, it faces the basement membrane. The apical surface is on the other side. It faces away from the basement membrane. And a lateral surface would be between the basal and apical surface, what you think of as a sidewall. Epithelial cells tissues are classified by the number of cell layers. There either is one cell or several cells. If it's a single cell layer, it's called a simple epithelia, and that cell is anchored to the basement membrane. If there's more than one cell, it's called stratified epithelium, and the cells are on top of the others, and they're not in direct contact with the basement membrane, and they don't get as much access to the nutrients as the ones on the basement membrane. So we have simple epithelium. These would be simple columnar. We have pseudostratified. This is still one layer. They all touch the basement membrane, but it looks like there are multiple layers. And here is a stratified epithelium with different layers of cells. We also talk about their shape. Squamous cells, cuboidal cells are roughly the same width as they are height, while columnar cells are taller than they are wide. So again, they're named by the shape. Simple squamous would be thin, scaly cells. Simple cuboidal are squarish, rounded, and simple columnar are tall, narrow. The other type is that pseudostratified columnar. All the cells don't reach the surface, but they all touch the basement membrane, so they appear stratified because some cells are taller than others. There are also goblet cells, which are shaped kind of like a wine glass. They are mucus-secreting cells. They're found in simple columnar and pseudostratified epithelium. So simple squamous epithelium, you'll find a single row of thin cells. This is places where we have rapid diffusion or transport of substance. They often secrete serous fluid. They're located in the alveoli, the glomeruli, the endothelium, and the serosa. Simple cuboidal, we have a single layer of round cells. They're usually more round than square, but they're about the same width as height. They're involved in absorption and secretion and mucus production, also in movement. They're located in the liver, the thyroid, the mammary, the salivary glands, the bronchioles, and kidney tubules. Simple columnar is a single row of tall, narrow cells. They have oval-shaped nuclei, usually in the bottom half of the cell, connecting to near the basement membrane. They often have a brush border of microvilli that we talked about before when we talked about microvilli. Sometimes they're ciliated. Many of them have goblet cells intermixed among them. They're involved in absorption and secretion, specifically secretion of mucus. We find them in the lining of the GI tract, the uterus, the kidney, and uterine tubes. Ciliated pseudostratified columnar epithelium, again, looks multi-layered, but all the cells are in contact with the basement membrane. The nuclei tend to be at different layers. They often have cilia and goblet cells. They are secreting and propelling the mucus along their surface. We find them in the respiratory tract and portions of the male urethra. Stratified epithelia range from 2 to 20 or more layers of cells. Some cells are directly on others, only the deepest ones attached to the basement membrane. So there are 3 stratified epithelium, and they're named for the shape of the cells, but it's the cells at their surface, the cells on the apical side. And these would be stratified squamous, stratified cuboidal, and stratified columnar. Uroepithelium is unique to the urinary tract and traditionally it's been called transitional epithelium, but the more correct anatomical term is uroepithelium because we find it in the urinary tract. Stratified columnar is rare. It's only found in places where two epithelial types meet. Stratified squamous is the most widespread. The deepest cells are cuboidal, maybe a little columnar. They include mitotically active stem cells, and as the daughter cells push upward towards the surface, they become flatter and migrate and finally die and flake off, a process called exfoliation. There are two kinds of stratified squamous epithelium. Keratinized or cornified, which is found on the skin, is very abrasion resistant, and we'll have most of our talk on the skin system is going to be about stratified squamous keratinized epithelium. Non-keratinized are found lining internal spaces. They don't have the surface layer of dead cells the way that the skin does. So they'll have multiple layers. They become flat and scaly towards the surface. They resist abrasion, retard water loss through the skin, resist penetration of organisms. They're found in epidermis, our palms, our soles. They are the most heavily keratinized regions. Well, the non-keratinized has same epithelium, but not the layer of dead cells. They're still good at resisting abrasion and pathogens entering. Find them in our tongue, our oral mucosa, esophagus, and vagina. Stratified cuboidal epithelium is two or more cell layers. Surface cells are square around. It secretes sweat, produce sperm, produce ovarian hormones. Find them in sweat glands, ovarian follicles, and seminiferous tubules. Uroepithelium, and I'm sorry, I still think of it as transitional epithelium, is a multi-layered epithelium with surface cells that change from round to flat when they are stretched. They're sometimes referred to as pillow cells. They allow for filling of the urinary tract, and we find them in the ureter and the bladder. Connective tissue is the most abundant, widely distributed, and variable type of tissue, in which cells occupy less space than the matrix, and a lot of the properties of connective tissue are defined more by the matrix. Most cells are not in direct contact with each other. There is a variability among vascularity. Some have, like loose connective tissues, have many blood vessels. Others, like cartilage, have no blood vessels. Connective tissues connect things. They bind organs, connect one bone to another, muscles to bone, skin to muscle, hold the organs in place. They provide support for the body and its organs. They form its internal framework. They provide physical protection, cushioning delicate organs, and they're also involved in immune protection as one of the connective tissues will be the location where those immune fighting cells develop. They're involved in movement. The bones provide a lever system. They're involved in storage. They have fat storage, calcium storage, phosphorus storage in the bone. They're involved in heat production. Brown fat generates heat. Transport. Blood transports gases, nutrients, wastes, hormones, and other blood cells. So, first type we'll look at is fibrous connective tissue. Fibrous connective tissues classified together because fibers are conspicuous components of this tissue. The cells themselves are fibroblasts, which produce fibers and ground substance that make up the matrix. Macrophages arise from monocytes, phagocytize foreign material, and activate the immune system when they sense foreign antigens. Leukocytes present, or white blood cells, function in immune defense. Plasma cells arise from lymphocytes and synthesize antibodies. Mast cells secrete heparin, inhibit clotting, and histamine to dilate blood vessels. Adipocytes, fat cells, are clustered in some fibrous tissues where they dominate, called adipose tissue. So these are all cells you would find within fibrous connective tissue. The fibers are going to be collagen fibers made up of collagen, which is a tough, flexible, stretch-resistant fiber. They are called white fibers because they truly do appear white in fresh tissue. They're going to make up things like tendons, ligaments, deep layer of skin. These are mostly collagen. They're less visible in the matrix of cartilage and bone, but they're still there. Other type of fibers are reticular fibers. Reticular fibers are very thin collagen fibers that are coated with a glycoprotein. They form a framework, especially in the spleen and lymph nodes, and they are part of basement membranes underneath the epithelial cells tissues we just talked about. The third type of fiber are elastic fibers. They're thinner than collagenous fibers. They're made of a protein called elastin, and like elastics, they allow stretch and recoil. So you can see why these are called white fibers. Other components of the connective tissue include ground substance, which is a featureless substance surrounding the cells, usually kind of a gelatinous or rubbery type structure. It absorbs compressive forces, has large molecules, GAG, proteoglycans, glycoproteins. Those GAGs are glucosaminoglands. They're a long polysaccharide composed of amino acids and uronic acid. They help to regulate water and electrolyte balance of tissues. Examples of these would include chondroitin sulfate, heparin, hyaluronic acid. There are proteoglycans, which are gigantic molecules shaped like bottle brushes. They form gravy-like colloids that hold tissue together. Then there are adhesive glycoproteins, which are protein carbohydrate complexes, and they're going to bind components of a tissue together. So fibrous connective tissue can be broken down into two large categories, loose and dense connective tissue. The loose connective tissue is mostly ground substance in the space surrounding the cells. Two good examples of this are areolar and reticular tissues. The dense connective tissues are mostly fibers in the space surrounding the cells. So mostly fibers or mostly ground substance. The dense can be dense regular or dense irregular. So areolar tissue is a loosely organized fibers. Abundant blood vessels possess all 6 cell types and fiber types. Find it underneath epithelia in the serous membranes, between the muscles, the passageways for nerves and blood vessels. Reticular tissue is a mesh of reticular fibers and fibroblasts. It forms supportive stroma, or framework, for lymphatic organs. We find it in lymph nodes, spleen, thymus, and bone marrow, all which are part of the lymphatic system. Dense, regular connective tissue is densely packed parallel collagen fibers that, with compressed fibroblast nuclei. The elastic tissue forms wavy sheets in some locations. Tendons attach muscles to bone, and ligaments hold bones together. Dense irregular is densely packed, randomly arranged collagen fibers with very few visible cells. And it's good for withstanding unpredictable stresses. So we find it in places like the deep layer of the skin and capsules around organs where you can't always predict where the stresses will come from, what direction. Adipose tissue or fat tissue is adipocytes, which are the dominant cell, and the space between the adipocytes is occupied by areolar tissue, reticular tissue, and blood capillaries. The fat is going to be the body's primary energy reserve. The quantity of stored triglycerides and the number of adipocytes are quite stable in a person. Fat is recycled continuously. New triglycerides are synthesized while old molecules are hydrolyzed and released to the blood. There are 2 types in humans, white or brown fat, white or yellow, and brown fat. White adipose tissue is the most abundant and significant type in adults. It's going to provide a lot of thermal insulation, going to cushion organs like our eyeballs and our kidneys, contributes to body contours. It's different in distributed differently in males and females, and it secretes hormones that regulate metabolism. Brown fat is found mainly in fetuses, infants, and children. Adults do have smaller deposits of it, though. The color comes from the blood vessels, the mitochondria, and the mitochondrial enzymes that are there, and it's going to function as a heat-generating tissue. So here's white adipose tissue, empty-looking cells with thin margins. The nucleus is pressed up against the cell membrane. That emptiness is really fat being stored in there. So it's for insulation, storage, cushioning, while brown fat would produce heat as well. Cartilage is a stiff connective tissue with flexibility and a rubbery matrix. It gives shapes to the ear, the tip of the nose, and the larynx. Cartilage has chondroblasts, cells that produce the matrix. Chondrocytes, cartilage cells that are trapped in lacunae or cavities within the cartilage. And pericardium, sheath of dense, irregular connective tissue that surrounds the elastic and mostly hyaline cartilage. We do not find that perichondrium in articulate cartilage. It contains a reserve population of chondroblasts that can contribute to cartilage, so it will grow throughout life. There are no blood capillaries. Cartilage is avascular. Diffusion is used to bring nutrients and remove waste. It heals very slowly. It's very rich in GAGs and contains collagen fibers. The types of cartilage vary with the various fiber compositions. We have hyaline cartilage, fibrocartilage, and elastic cartilage. Hyaline cartilage is a clear, glassy look under the microscope because of the fineness of the collagen fibers. It eases joint movements, holds airways open, moves vocal cords, and it's important in the growth of long bones in juveniles. It's located in locations include articulate cartilage, costal cartilage, trachea, larynx, and the fetal skeleton. Elastic cartilage contains an abundance of elastic fibers. It is covered with perichondria, provides flexible elastic support. We find it in the external ear and the epiglottis. While fibrocartilage contains large, coarse bundles of collagen fibers, it's very good at resisting compression and absorbing shock. We find it in the pubic symphysis, the menisces, and the intervertebral discs. Bone, or osseous tissue, is a calcified connective tissue that makes up the skeleton. It comes in two different forms, spongy bone and compact bone, and all the bones we think of, the organ bones, contain some ratio of spongy and compact. The spongy bone is a delicate slivers and plates given a spongy appearance. It's found in the head of long bones, in the middle of flat bones like the sternum, while compact or dense bone has none of those visible spaces. That's going to form the external surface of all bones. And when you typically think of a bone, you're thinking of the compact form of the bone. Compact bone is arranged in cylinders called osteons. There's a central canal, a passage that runs longitudinally along the bone shaft and contains blood vessels and nerves. It's made up of concentric layers or concentric lamellae surrounding the central canal. An osteon is a central canal and its surrounding lamellae. Osteocytes are mature bone cells within lacunae between the lamella, and caniculi are channels that radiate from each lacuna to its neighbors, allowing the osteocytes to maintain contact with each other. Periosteum is a tough, fibrous connective tissue covering the whole bone. Here you see those layers of osteons, the cylinders that surround the osteonic canal runs longitudinally down the shaft of long bones. Blood is a fluid connective tissue travels through tubular blood vessels, transports cells and dissolved material from place to place. It contains formed elements, but those formed elements are suspended in a liquid ground substance that we call blood plasma. The formed elements include erythrocytes, which transport oxygen, leukocytes, which defend against infectious diseases, and platelets, which are fragments involved in the process of clotting. So they are called formed elements because they are cells or portions of cells. They are not, the erythrocytes and platelets are not true cells. So erythrocytes appear as a pale pink disc with light centers. Leukocytes are slightly larger and have various shaped nuclei. Platelets are very small cell fragments and they do not have a nuclei. Nervous tissue and muscular tissue are excitable tissues, which means they respond to stimuli, and they do so by changing membrane potential. It's developed to its highest degree in nervous and muscular tissue, so they are often referred to as excitable tissues. Excitation founded in charge differences, which we would know as voltage across the membrane, and we call that membrane potential. In nerve cells, the changes in voltage result in rapid transmission of signals to other cells, whereas in muscle cells, those changes in voltage result in a contraction or shortening of the cell. Nervous tissue is specialized for communication by electrical and chemical signals. It consists of neurons, the cells that detect stimuli, respond, and transmit coded information to other cells, and glial cells, or neuroglia, which are the housekeepers, the protecting and assistants to neurons that make up actually the bulk of the nervous system. A neuron has a cell body, which has a nucleus and other organelles and controls protein synthesis. It has dendrites, which are branch processes that receive signals from other cells and deliver them to the cell body, and an axon, which sends an outgoing signal to other cells. It can be very long, up to a meter, and although there's only one axon generally, it can make contact at its end with several different target cells. Muscular tissue is specialized to contract when stimulated, exerting a physical force on other tissues, organs, or fluids. It's also an important source of body heat. Muscle tissue comes in three types: skeletal, cardiac, and smooth. Skeletal muscle makes up our voluntary muscular system. It's made of muscle fibers, which are long, thin cells. Most of the muscles attach to bone on both sides. They contain multiple nuclei adjacent to plasma membranes. They have striations. You see the little lines in the images, alternating dark and light bands, and they are under voluntary or conscious control to contract. Now, that doesn't mean they don't have some reflexes that occur, but you can consciously control the contraction of skeletal muscle. Cardiac muscle is only found in the heart wall. The cardiomyocytes are short, branched, with one centrally located nucleus. There are intercalated discs that join the cardiomyocytes end to end. It is striated, much like skeletal muscle. However, it is involuntary. It is not under conscious control. The cardiac muscle has its own system for firing. It can be influenced by the nervous system, but it has its own intrinsic beat mechanism. Smooth muscle are short, fusiform-shaped cells. Cells have one central nucleus. They do not have striations. They still use actin and myosin, but their organization is different in smooth muscle. They are also involuntary, and most of our visceral muscles make up parts of the walls of hollow organs. Now we have junctions, glands, and membranes. Cellular junctions are connections between two cells. Most are anchored to each other on their matrix. The cells communicate with each other, resist mechanical stress, and control what moves through the gaps between them. Tight junctions are zipper-like interlocking linkage between two adjacent cells by a transmembrane cell adhesion protein. Will encircle an epithelial cell near its apical surface, joints the epithelial cell with the neighboring cells, seals off intracellular spaces, making it difficult or impossible for substances to pass through the cells. So we have tight junctions in the stomach and intestines to prevent digestive juices from getting out of those lumen and into the underlying tissue around them. Desmosomes are more like a patch that holds cells together, sort of like clothing snap. They keep cells from pulling apart, so they resist mechanical stress. They have a hook-like J-shaped proteins that rise from the cytoskeleton. They're very common in the epidermis and other epithelium. A hemidesmosome is sort of a half desmosome that anchors the basal cell of an epithelium to an underlying basal membrane. So epithelium cannot easily peel away from the underlying tissue. And a gap junction is a channel between cells formed by a ring-like structure called a connexon. So the connexon consists of 6 transmembrane proteins arranged in like a segment of an orange around a water-filled channel. So ions, nutrients, and other small solids can pass directly between the cells. Find these in cardiac muscles, where we call them intercalated discs. They're also found in smooth muscle, embryonic tissue, the lens and cornea of our eye. Glands are cells or an organ that secretes a substance for use somewhere else in the body, usually composed of epithelial tissue and a connective tissue framework and capsule. They are involved in secretion, the product of something useful to the body, or excretion, removal of a waste product. Glands are classified as either exocrine or endocrine. An exocrine gland maintain their contact with the surface of the epithelium by way of a duct. So the surfaces can be external, sweat glands, tear glands, or internal. We have them in pancreas and salivary glands. Endocrine glands have no ducts, but many blood capillaries, and they secrete their hormones directly into the blood. A hormone, if you don't know, is a chemical messenger that stimulates cells elsewhere in the body. Thyroid, adrenal gland, pituitary gland are all examples of endocrine glands. Unicellular glands are secretory cells found in an epithelium that is predominantly non-secretory. They can be exocrine or endocrine. Mucus-secreting goblet cells in the trachea or endocrine cells in the stomach are examples. Exocrine glands have a capsule, connective tissue covering the exocrine gland, a septa or trabeculi, the extensions of the capsule that are going to divide the interior of the gland into large compartments we would call lobes. Finer septa may divide those lobes into microscopic lobules. Stroma is connective tissue framework of the gland. It supports and organizes the glandular tissue. Parenchyma are cells that perform the task of synthesis and secretion. These are typically cuboidal or simple columnar epithelial cells. Exocrine glands are classified by the structure of their ducts and their secretory portions. So for duct shape, we have simple or compound, unbranched or branched. And for secretory portions, we have tubular, acinar, or tubulo-acinar. The tubular are narrow secretory portion, duct, and secretory portion that are uniform in diameter. Acinar, the secretory cells form a dilated sac, and in the combination we have secretory cells that are both in tubular and acinar portions. And those are shown here. Glands are also classified by their secretion. We have serous glands, mucus glands, and mixed glands. Serous glands produce a thin, watery secretion, perspiration, milk, tears, digestive juices. Mucus glands produce a glycoprotein called mucin, which absorbs water and forms mucus. Goblet cells, we've talked about numerous times today, are unicellular mucus glands. Mixed glands have both serous and mucus cells and produce a mixture of the two types. So 2 pairs of salivary glands in the chin would be examples of these. Exocrine glands are classified according to how their secretions are released. We have eccrine or merocrine glands, which release their products by exocytosis. Tear glands, pancreas, gastric glands. Apocrine secretion, the lipid droplet covered the membrane and the cytoplasm buds from the surface of the cell. This is a way that milk fat is secreted by mammary glands. And holocrine secretion, the cells accumulate the product and the entire cell will disintegrate. That secretes a mixture of cell fragments and synthesized substances. Oil glands of the scalp, the skin, and glands of the eyelids are holocrine glands. Membranes may be only epithelial, only connective, or a mixture of epithelial, connective, and muscular tissue. So a cutaneous membrane is the skin. It's the largest membrane of the body. It's composed of stratified squamous epithelium resting on a layer of connective tissue. It's dry and it serves a protective function. Mucous membrane line the passages that open to the external environment, digestive tract. They have sublayers, epithelium, lamina propria, and muscularis mucosa. They have an absorptive, secretory, protective function, and often have goblet cells, as shown here. A serous membrane lines cavities of some internal body cavity. There's simple squamous epithelium. It's called mesothelium. Rests on a layer of areolar tissue. Produces serous fluid that arises from the blood. It covers the organs and lines walls of the body cavity. Pleurae, pericardium, peritoneum, for example. Tissues grow by increasing the number of cells or the size of the cells. Hyperplasia would mean you're growing through cell multiplication. Most embryonic and childhood growth would be hyperplasia. Hypertrophy would be the enlargement of pre-existing cells. That's how we get muscles to enlarge through exercise. Also accumulates body fat. Neoplasia is the development of a tumor, a neoplasm, composed of abnormal, non-functional tissue. It can be benign or malignant. Differentiation is the development of a more specialized form and function from unspecialized tissue. So embryonic mesenchyme will eventually become cartilage and bone. It will differentiate into that. Metaplasia is changing from one type of mature tissue to another. So simple cuboidal tissue of the vagina before puberty changes to stratified squamous after puberty. Pseudostratified columnar epithelium, the bronchi of smokers, changes to stratified squamous epithelium. Stem cells are undifferentiated cells that are not yet performing any specialized function. They have the potential to differentiate into one or more types of mature functional cells. There is developmental plasticity, which is the ability of a stem cell to give rise to a diversity of mature cell types. They are isolated from the Morilla stage of human development, or embryonic stem cells. And actually, stem cells we classify as 2 different types. Embryonic stem cells, they come from the early human embryo. Initially, they are totipotent. We all started out as one totipotent embryonic stem cell. Eventually, most of them become pluripotent stem cells where they can develop into specific things. And then the other type of stem cells are the adult stem cells. They're undifferentiated cells found within organs. Some of them are multipotent. The ones for the blood can develop into any blood cell. Many are unipotent. They can only become one cell type. skin stem cells can only become skin cells. Damaged tissue can be repaired through regeneration or fibrosis. In regeneration, we replace the dead or damaged cells with the same type of cells as before. It should restore normal function. That happens all the time in the regeneration of our skin, in our liver. In fibrosis, we replace damaged cells with scar tissue. That preserves the structure of the organ, but it doesn't bring back the function. Severe cuts and burns are repaired this way, scarring of the lungs and tuberculosis. So the vessel will leak blood into the cut. Mast cells and other damaged cells will release histamines that dilate blood vessels, make capillaries more permeable. Plasma seeps into the wound. It carries antibodies and clotting proteins that will help to recruit more. So the blood clot will then knit the edges of the cut together and inhibit the spread of any pathogens that may be there. It will form a scab that seals the wound and blocks the infection. Then macrophages will come in and phagocytize and digest the tissue debris. New capillaries begin to sprout from nearby vessels. Deeper portions of the clot become infiltrated by capillaries and fibroblasts, will transform into a soft mass called granulation tissue. Macrophages will remove the blood clot. Fibroblasts will deposit new collagen, fibroblastic, the reconstructive phase. This begins 3 to 4 days after injury and usually lasts up to 2 weeks. Epithelials around the wound will multiply, migrate beneath the scab. The epithelium will regenerate. The underlying connective tissue undergoes fibrosis. Scar tissue may or may not show through the epithelium. So we get a remodeling or maturation phase that begins several weeks after the injury and can go up to 2 years. Atrophy is the shrinking of tissue through a loss in cell size or number. It happens because of aging and a lack of use. Necrosis is a pathological tissue death. An infarction is the death of a tissue because the blood supply, oxygen supply, is cut off. In the case of gangrene, tissue necrosis is due to insufficient blood supply, generally linked to an infection or an injury. Dry gangrene is a common complication of diabetes. Decubitus ulcer or bed sores are pressure sores that become dry gangrene with continual pressure on the skin. Wet gangrene is a liquefaction of internal organs with infection. And gas gangrene is usually from an infection of a soil bacteria, Clostridium, that results in hydrogen bubbles within the tissue. None of the gangrenes are good. And here's one in foot as a complication of diabetes mellitus. A different type of cell death is called apoptosis. It's a programmed cell death. This is a normal cell death. Cells that have completed their function, and the best thing for them to do is to disappear. They become phagocytized by macrophages and other cells. We could call it a built-in suicide program. This does not lead to inflammation. And that brings us to the end of our discussion on tissues. All right,